All right. Hello, everybody. My name is Kaitlyn. Like Dan said, I'm in the Brockerhoff lab. I'm a graduate student. And today I'm going to be talking about my project, which is trying to figure out why mutations in IMPDH1 is causing autosomal dominant retinitis pigmentosa and LCA. And we have a couple of zebrafish models to address this. And I think there's some interesting cellular and metabolic defects that I'd like to go over today. But before I get into the presentation, just want to make known that please stop me at any point if you have any questions or comments. I'm happy to pause and chat about anything. Okay. All right. So I'm going to start with these fundus images. Here is a normal eye, and here is an eye that has from a patient that has an IMPDH1 mutation. And it looks similar to a retinitis pigmentosa phenotype, where traditionally in RP, you have narrowing of the peripheral field and then secondary cone loss. There's a ton of different proteins that can lead to RP and LCA, and IMPDH is one of those that was discovered in 1993 to be associated with photoreceptor loss. IMPDH mutations are a little bit different from the traditional RP mutations in that they seem to affect both rods and cones, which makes sense because they're both robustly expressed in both rods and cones. So we are fortunate enough at UW to be next to the Sebastian lab, who has an adaptive optics scope. And so what you're looking at in these images are human patients, and each of these are individual photoreceptors, in this case cones, because they're imaging near the fovea. And so what they are showing in this graph is cone density at different degrees away from the fovea. And if you look at a control patient or a rhodopsin mutant patient, you see cone preservation near the fovea, and it gets lessened as you go away from the fovea. However, IMPDH is a bit different because we're seeing cones have degenerated in the fovea and are just generally lesser. And this agrees with some other data that shows an early cone ERG deficit with these mutations. And so we think that it's impacting both rods and cones. And so to take a step back, let's talk a little bit about what IMPDH is. So it is a rate-limiting enzyme in the de novo purine biosynthesis pathway. It is found in low levels in most cells, and again, robustly expressed in rods and cones. And if we think about why purine biosynthesis is important generally in photoreceptors, the first thing that comes to my mind is cyclic GMP. It's important in phototransduction and can be produced through this pathway. Additionally, photoreceptors require a ton of ATP, and this is a way to generate ATP as well, as long with just needing nucleotides for RNA and mitochondrial DNA. So perturbations in any of these may be causing problems that is detrimental to cone health. And so if we look at this pathway, IMP can be converted to XMP with IMPDH. Like I said, this is a rate-limiting reaction, and to get this, NAD is converted to NADH. XMP can then be converted to other useful products like GTP and cyclic GMP. If IMPDH is inhibited, though, IMP can be shunted toward this side of the pathway that can produce ATP and other associated products. So you can imagine that having good regulation of this enzyme is important for these nucleotide pools. And so, although the Brockerhoff lab is a structural lab, we collaborate really heavily with the Coleman lab, which does look at IMPDH structure. So I'm going to spend a little bit of time talking about the structure because it's important for its function. So if we look at IMPDH in its monomeric form, it has two domains, this regulatory domain in pink, where ATP and GTP can bind. And in green here is the catalytic domain where IMP and NAD+ can bind. These monomers connect together to form a tetramer that stack on top of each other to form octamers. And the conformation of these octamers changes based off of the nucleotide status of the cell. So if ATP is bound to two of these sites here, the enzyme can adopt an extended conformation and is more active. Whereas if GTP is bound to two or three of these regulatory sites, the enzyme can compress and be inhibited. And these octamers here can actually stack on top of each other to form these long, beautiful filaments. And the filaments themselves are more active than these octamers. So it's again another way to regulate enzyme activity. So that's what it looks like structurally. What does it look like in a cell? So this is a cell culture that has, excuse me, IMPDH fluorescently marked. And we can see this nice filament here that is dynamic. It's moving and changing some of its shape. So that's what it looks like in cells. But back to the retina. So I mentioned that several mutations can lead to autosomal dominant RP or LCA. There are a ton of different IMPDH mutations that can lead to this phenotype. And a lot of these are in the regulatory domain, which is where ATP and GTP is binding. So that's kind of providing us a little bit of a clue of what could be going on, including one of the most common mutations that I'll be talking about later in my talk, found in the human population is in this region. So like I said, IMPDH is found at low levels in most cells and expressed really robustly in the retina. And the retina, in fact, has its own special IMPDH variant. And the main difference here is that the retinal variant has these N and C termini. And this is really important to help this enzyme adopt a conformation in between the completely inhibited and active state. So at high levels of GTP, instead of becoming completely inhibited, it locks it in this flat interface where it can remain partially active. And so in our lab, we work with zebrafish. We wanted to make sure that the IMPDH is conserved, and especially in these N and C termini, and we do see good conservation between zebrafish and human IMPDH1. Additionally, we wanted to look at it structurally with the Coleman lab. So we took zebrafish IMPDH1, and we can see it forms really beautiful filaments in this negative stain EM. And we also have a engineered mutant, this Y12A, that isn't seen as a disease-causing mutant, but it's a really nice way to break this filament assembly to study, the impact of IMPDH1 being in a filament. And so an additional thing that the Coleman lab can do really well is measure IMPDH activity in vitro at different concentrations of GTP. And so if we just take the canonical IMPDH without the N and C termini, we can see that at a certain concentration of GTP, it becomes inhibited. However, the retinal variant is able to stay active at higher levels of GTP, but eventually again will become inhibited. And if we engineer this mutation that breaks filament assembly, we lose that GTP sensitivity or inhibition sensitivity. And so perhaps IMPDH being in a filament erroneously can cause it to be more active when it shouldn't be. And so now to get to what this looks like in the eye. So we work with zebrafish, which I mentioned before, and we wanted to just see what IMPDH looks like in a wild type zebrafish without any mutations. And so here we started by looking at the rods, which are in magenta here, and we can see IMPDH in green, and it's these little filaments throughout the rods. This is a similar, story in cones. So here are zebrafish cones. The mitochondria this time are in magenta. And again, IMPDH is found throughout the cell body, some by the mitochondria, some in the synapse, these little small filaments. And in a couple of cases, they're hanging out in the outer segments. And so we know that mutations cause photoreceptor degeneration. To more characterize the importance of IMPDH1 in the retina, we wanted to see what happened if we knocked it out. And immediately we wanted to know about its metabolism since it's a metabolic enzyme. And so we collaborated with the DO lab to look at steady state metabolism of the knockout. So if we knocked out IMPDH, we'd expect this side of the pathway to be reduced. And indeed, that's what we see. We see a reduction of GMP to IMP, but this side of the pathway is not impacted. We also looked at several other metabolites, and we can see that guanine is significantly reduced. It's about 50% less when IMPDH is not around. That's not the case with all of these metabolites, but we do see a clear difference in guanine. And notably, we don't see a difference in cyclic GMP, which was pretty surprising. Seems that those levels are okay, even when IMPDH is not around. And so with this metabolic phenotype, we wanted to know if there were going to be any structural changes in cone morphology. So we turned to confocal microscopy to look at the structure of cones. And so here we have the cone cytosol in green here. In a wild type fish, of course, they look beautiful even at 18 months, which is quite old for a fish. And in the knockout model, we see preservation of cones. And if you look above the cones, you can see that the rods are also still there, and you can look at the rod nuclear layer. So we are not seeing any structural changes or any signs of cone degeneration by looking at them. We also turned to electron microscopy and saw that, again, we didn't see any major changes looking at EM at how these photoreceptors look between the wild type and the knockout. And so this really got us curious because it seems to be better to have no IMPDH around in the retina than a mutated version. We really want to understand why that is and what could be happening with these mutants. And so to take a step back and look at these mutations more broadly, the Coleman lab was able to characterize some of them into two different classes. So these class 1 mutants, when they did their activity assay where they measured IMPDH activity, again, at a certain level of GTP, it will become inhibited. But with these class 1 mutants, they seem to be active at very high levels of GTP. And at the levels that we tested, they weren't getting inhibited at all. And these are highlighted in red here. And then on the other side, we have some class 2 mutations that don't have a change in their GTP inhibitions, highlighted in blue. We wanted to start out kind of attacking these mutants with the class 1 mutant, which seemed to have a clearer phenotype. And so we generated 2 zebrafish models, a D226N model, which is the most common IMPDH associated mutation, and K23AD. And this was pretty exciting because these are the first IMPDH mutant animals to study the eye. And so with these fish, our hypothesis was, or is, that with the class 1 mutants, photoreceptors are degenerating because this enzyme is active when it shouldn't be. And so to generate the fish, we injected them with our IMPDH mutated or mutant IMPDH DNA with a MIC tag, so we can tell the mutant IMPDH from endogenous. It's under a cone promoter, and then we have a red heart marker so that we can easily screen them. We inject them at the 1 cell stage, and then we grow them up a couple of generations so that we have a stable line. And then once we have a stable line, we collect eye samples when they are larvae to check expression levels. So with K23AD, we have about a 2.6 to 1 mutant to wild type. D226N is about 2.3 to 1. So it's a little higher, but it's not a 100x overexpression. We're trying to keep it as close to endogenous levels as we can. And then as a control, we injected just wild type IMPDH to bring those levels a little bit higher at 1.3 to 1 to make sure that the overall protein expression isn't causing phenotypes. And then we grow up the fish and use the adult fish for experiments. And so our first question does this recapitulate human disease? Do we see any signs of degeneration? So here's just a wild type fish, about 4 months old. You can see the cone nuclear layer here. It's looking very nice. K23AD at 4 months, we're starting to see very severe degeneration of those cones. And I was, again, concerned about the expression level of IMPDH. So I grabbed a fish with really low expression. So this is about 0.53 to 1 mutant to wild type. And again, I see very severe degeneration. Doesn't seem to be the level of transgene expression causing this phenotype. D226N, on the other hand, degenerates much later. We haven't seen a full degeneration yet, but we're thinking that this doesn't look too healthy, and we're in the process of quantifying it. And then the wild type, or the transgenic wild type, seems to be fine. This is a 4-month image, but I've checked later times, and I don't see any signs of degeneration. And so our big question here is, why are these degenerating? So the first thing that we looked at was the filaments themselves. So this is a wild type zebrafish. There's no mutant or transgenic IMPDH, so there's no mixed signal here. It's just endogenous IMPDH in red, and the cones are in green. So you can see, again, there's some by the synapses and throughout the cell body. When we looked at K23AD, we're starting to see these, what appears to be massive IMPDH filaments in the synapse, and not so much of IMPDH filaments in the cell body by the mitochondria, etc. D226N was a similar story. Again, we see this massive response at the synapse and some filaments hanging out near the nuclei as well. And then finally, our transgenic wild type, where it's just mostly cytosolic signal, and we're not seeing those huge filaments. But looking at these images, we wanted to get a little bit better resolution, because it's really hard to see what's going on in the synapse when just so much of it is there. And so we turned to our vision core, which can do expansion microscopy. So we took a retinal section and we expanded it about 4x. And then this is a stitched image, and we can get really better resolution here at the synapse and see what it actually looks like. So here is a close-up of a IMPDH filament in the nuclei of these cells, and then in the synapse, where instead of just kind of a massive signal covering all of it, we're seeing these huge IMPDH bundles of filaments that are even starting to curve into these like C shapes. And so we wanted to visualize this in another way and look more at what is going on in the synapse. So we turned to EM, and here's just a wild type zebrafish image. We can see the synapses here, the vesicles and some ribbons. And then we looked at our D226N, where again, we see vesicles and ribbons. So crudely, the machinery seems to be there, but we see these really long filamentous structures that we think are IMPDH, because we don't see them at all in the wild type.
Sorry, you don't see them at all in the wild type overexpressor as well?
We haven't checked that, but I just submitted eyes to look for that.
Okay.
Yeah, good question. Okay. So we know that it being in a filamentous form, the enzyme is more active. So we really are curious about what's going on metabolically. So we started with steady state metabolism to see if there's any perturbations in the pools. And so we hypothesized that if IMPDH is active, overactive, then we would see an increase in the cyclic GMP pool sizes. And what we don't see is that, so we see that there isn't any significant difference between these two mutants or the transgene wild type. And so this doesn't seem to be an impact. However, this is steady state metabolism when we're really curious about what the flux will look like when we get to it. So we wanted to look at other metabolites as well. So again, we would expect this whole side of the pathway to be up. And so looking at this, we see that K23AD actually has a decrease in the GMP to IMP ratio and also in the AMP-IMP ratio. So it's as if both sides of these pathways are being shut down. And D226N, on the other hand, again, another class 1 mutant, has a very different phenotype. We're not seeing any changes here. And notably, this change is distinct from the knockout. So we still think this is a gain of function mutation. And so we wanted to look at other metabolites in purine and pyrimidine pathway. And for K23AD, see similarly that cyclic AMP is down, AMP is down. Some of these are trending down. Hopefully with a higher N, we can get better clarity if this is actually significant or not. And for D226N, we don't see any changes in the purines per se, but we do see changes in pyrimidines. And so we're starting to think that IMPDH could be interacting with other proteins and either trapping them or moving them to a place where they're not supposed to be. And then our transgenic wild type, we're not seeing any changes in the purine or pyrimidine metabolites. And so this lends to our new hypothesis, which is, does IMPDH interact with other metabolic proteins? So with K23AD, we saw a reduction in both the GMP side and the AMP side. So perhaps IMPDH is interacting with ADSS and moving it or trapping it to where it's not functional. Additionally, for D226N, it could be interacting with CTP synthase, which would explain the pyrimidine perturbation, and PRPS, which is further up in the pathway. These are just a short list of some of the ones that we're thinking about. And it's not completely out of left field. There's been some studies that have shown that IMPDH is known to interact with other metabolic filaments, like CTP synthase, for example. This is done in cell culture where CTP is in red here, and IMPDH, you can see a really nice colocalization. We're not sure if this holds up in the retina, but I wouldn't be surprised if in these massive filaments, we're seeing other proteins getting trapped there as well. And so the final thing that I'll talk about is looking at the mitochondria. And the reason why we went here is because if there is an imbalance in nucleotides, it could impact the mitochondrial DNA or RNA. And so to orient you to just wild type zebrafish mitochondria from cones, we have the cone outer segment up here, and the mitochondrial cluster is in this pretty tight bundle right underneath the outer segment. And in wild type, they're usually pretty circular, although they can get kind of complex. And there's usually smaller circular mitochondria kind of forming like a bowl around the bottom of them. With the D226N mutant, however, we saw evidence of these more fused mitochondria. They're kind of hot dog in shape. So maybe that perturbation in mitochondrial DNA or RNA is kind of causing them to fuse together. We have repeated this, and it does seem to hold up, and we see in general that the D226N mitochondria look abnormal. So we're really excited to get more quantitative with the data set. It's a relatively new data set, and also look at the K23AD mutant as well. And so in general, what I hope to have conveyed is when I think about IMPDH mutations, I think that it could be causing degeneration with these IMPDH filaments not being where they're supposed to be and kind of aggregating together, which could be taking other metabolic proteins down with it. That could be impacting its metabolism, not only in the purine side, but also the pyrimidine side in some cases. And perhaps having a nucleotide deficit is causing mitochondrial defects as well. And so in the future, we really want to test if it is interacting with metabolic proteins and if that's perturbed in our disease model, get into metabolic flux experiments. All of the research that I explained today was steady state, and looking at mitochondrial DNA changes as well. And so with that, I'd like to thank my lab, especially Sue, a technician in the lab, Matthew, and undergrad Riddy, who've been really instrumental for the project. Whitney, who did a lot of the foundational work in the knockout project. The Coleman lab does our structural work. The Vision Core, Michelle for the expansion, and Ed for the TEM. And the Do lab, Jinhai, Mark, and Kevin, who have done a lot of work with LCMS. And with that, I can take any other questions.
I can ask a little question.
Yeah, of course.
So in zebrafish, I'm not a terrible expert on this, I have to say, but I was under the impression that there's always 4 alleles of a given gene. Is that true for IMPDH1 as well? And did you knock them out, all of them, or how does that work?
Yeah, so there are 2 versions of IMPDH, IMPDH1 and 2. We found that 1 is more robustly expressed, and then there's different splice variants of IMPDH1. We found 4 major splice variants, and we tested to see which one was most abundantly expressed. And then when we knocked out the IMPDH1, we made sure that there wasn't an overcompensation of IMPDH2.
Okay.
Yeah, and I can just add, there's a 1A and a 1B, and 1B and 2, they're really not expressed at significant levels in the retina. So it was
Okay.
kind of fortuitous in that not only is it 1A, but it's a specific splice variant of 1A that's in rods and cones. Which is great, because then the knockout is really kind of rod and cone specific.
Yeah. Thank you. Thank you.
So, do you think that the abnormal mitochondrial structure in cones in the mutant variant, is that linked to a deprivation of the IMPDH product or the CTP synthase product, or both? I guess do you have a guess?
Just based off of steady state metabolism with that particular mutant, the pyrimidine seemed to be impacted the most. And so if I was just looking at that data, that would be my guess. But that being said, we haven't done metabolic flux. And so that could, give us a whole nother, range of answers here.
Yeah. Well, I couldn't help but notice that the IMPDH in your wild type overexpressors seemed right outside the mitochondria, at least some of it, in addition to being at the synapse. And it did not seem true in the mutant overexpressors. So if we take that wild type overexpressor as a control, then, and we say perhaps there's, that diffusion of, of GMP or whatever is impaired, from the synapse to the, it's not rapid enough from the synapse to the mitochondria, and then perhaps there's just a local difference in concentration.
Right. Yeah, that could definitely be true. I could definitely see that. It will be really interesting to get the TEM back from the transgenic wild type to just see how their mitochondria look. Because I agree, having IMPDH around mitochondria is probably very important. We see it in the wild type without any transgene. And so I imagine it's serving an important function for the mitochondria.
Yeah, I just have a quick question about the knockout animals. Did you see any other phenotype outside of the eye with those?
I don't recall any. They seem to survive well. I haven't noticed anything grossly, but we haven't done any systematic study, look at other organs or anything.
And this is just like a speculative follow-up, because I worked with zebrafish in a past life, and when we knocked out genes, sometimes there'd just be no phenotype at all. And then my question always is why did the fish maintain these genes through evolution if they're apparently dispensable for their development and survival? What are your thoughts on that?
Yeah, that's a good question. I mean, it certainly is important for guanine, but it was really surprising that the cyclic GMP levels were okay. I'm not sure if it's just photoreceptors have evolved different ways to maintain that because it is such a critical part of phototransduction. But it is really curious to me. I'm not sure.
So, if you still have in the knockout basically like the same levels of cyclic GMP, right?
Yes.
And so where does that come from?
I imagine the salvage pathway.
And so
Working. Go ahead.
I guess if you apply stress to the salvage pathway, do you think you would start to see a phenotype in the knockout, or is that something that's been tried?
Yeah, we could. I'm trying to recall if there's any inhibitors or anything of the salvage pathway. I'm not sure.
Like
Yeah, that's
starvation.
interesting. Yeah, right. We haven't done any like long-term starvation, but that would definitely be interesting.
Yeah, that seems like a great idea, because it was surprising that 50% of the guanine in the knockout was well, like 50% was gone. So it was surprising to us that actually de novo synthesis played such a big role, since the cells aren't dividing. So I do think that if we stress them in some way, we might see a phenotype. Yeah.
That maybe goes to also address Eric's question. I don't know.
Yeah, absolutely. We just looked at happy fish in the fish facility with lots of food.
Yeah. I mean,
Yeah.
it applies to me too. I look at happy mice a lot.
Yeah.
I don't really want to make them unhappy, but sometimes we have to.
But the other thing that's curious about like the nucleotide not getting to the mitochondria is that the knockout didn't show the same phenotypes as the transgenics, right? So it's definitely a gain. It's something that the transgenes are causing, that the mutations are causing.
That's a good point.
So it's not just the loss of the purines, right? Or at least not at the guanine level. It could be the for K2380 that it's both adenine and guanine.
Okay.
And oh, sorry.
No, I
Okay.
was going to ask if there are any other questions. Go ahead.
Can you go back to the CTP synthase co-localization figure?
Yeah. Oh, there we go.
And so the yellow, that's the overlap, right?
Yes.
I was just trying to figure out whether so the CTP synthase also polymerizes, right?
Yes.
And I was wondering whether they're like separate polymers that are intertwined or something, but you can't really tell from this this figure, I guess.
Yeah, I'm not sure. With fluorescent microscopy, I don't think we could tell. I don't know if they've done any EM, but that would, to see like how they mix together.
It does kind of look like they're in pairs. The IMPDH filaments, you know, they seem to parallel each other, kinda, almost, if you squint. I don't know.
I was thinking, I wonder if the IMPDH is sort of on the periphery of a CTP synthase core or something, but it's probably, that's just fantasy, probably. I can't really tell.
Yeah, I haven't looked at the CTP synthase structure very closely, but that would be interesting. See how they interact.
Kaitlyn, this is Jason Miller. As someone who pays almost zero attention to purines and pyrimidines, and therefore needs to be well educated, can you give? I think the IMP levels did not change in any of these in these mutants, correct?
Mm-hmm.
So and obviously you presented this simplified pathway that they can be shunted over to the purines versus pyrimidines. What's known about and IMP is a sort of central point for all the nucleosides regulation. But what's known about how tightly the cell sort of keeps those levels tightly regulated? And is it surprising that the IMP levels didn't change at all, especially given that other things also didn't change, like you said, like cyclic, GMPs, et cetera? Or is that a not surprising feature that, when you push on one part of the pathways coming off IMP, there's just always compensation so that you maintain a really steady state level of IMP?
Yeah, it was certainly surprising to me because I would figure the buildup has to be somewhere if it's not going down that pathway. I thought it might be IMP, but maybe it's higher up in the pathway, even pentose phosphate pathway, which we didn't see with this round of LCMS, but we could certainly look into that pathway and we're curious about it. In terms of general regulation of ATP and GTP, from the small literature search I've done, I think it's just really cell type dependent, and some cells, just have various levels. I'm not sure if in the retina it's been extremely well characterized how tight those pools are and how they change in different contexts.
Yeah, I think Kaitlyn's flux experiments, once they get started, are going to be incredibly insightful regarding like how metabolites go through these different pathways and how it's regulated in photoreceptors.
Yeah, and all the way back to like what's happening in pentose phosphate,
Right.
in
Exactly.
these,
Right.
all, yeah.
Right.
All right.
Just a quick, wait, sorry, Dan, just quick, because it reminded me, a quick technical question. How are you guys going to do the flux in zebrafish? Are you going to anesthetize and IP them or feed them like the plankton that was raised in C13 glucose? It's just, and it's easy for mice to think, but zebrafish are just kind of a different animal.
Yeah, so we're currently working out the logistics of that. There was a paper published where you can feed them C13 in the water and they swim around in it. That would be really cool to do. I haven't seen anything with heavy labeled glycine, which is the fuel that we would use. We could always try. In general, when we do ex vivo, we just isolate the eye or the retina, depending on the age of the fish and how big it is, and then incubate it and then flash freeze and send it.
Very cool. Thank you.
Yeah.
I'm just on the logistics of it, swimming around in the water mean like a very costly amount of C13 label, or you can like get them down to a tiny bit of water?
Yeah, I haven't done that calculation, but it's probably pretty critical. Since the K238AE degenerate much faster, they might be a better one to do because they are so much smaller and they could just swim around in much less water.
You put them in a little Eppendorf tube.
Yeah, exactly.
Do you have to use adults for that, or do you think you get away with larvae and just dissect the retinas out when they're really young?
I'm not sure about larvae. For all of the metabolism I've done so far with K238E, they've been about a month old because they degenerate so fast. And the eyes are pretty small. Unfortunately, about 2 to 3 months, we can't dissect out the retina. So that's the big caveat there is ideally we want less cell types, but smaller. Yeah, it's challenging.
Thanks, Kaitlyn.
Yeah, thank you.
All right, and thanks to everyone for coming. I'm going to start posting these recordings on the Google Calendar that we have, or the Google sheet that we have for the calendar for these seminars. So this one will go on there later today, and all the other earlier ones I'll try to do that too. So thank you all for coming.
Thanks, Kaitlyn.